Chapter 13 Photosynthesis in Higher Plants. All animals, including human beings, depend on plants for their food. Have you ever wondered from where plants get their food? Green plants, in fact, have to make or rather synthesize the food they need, and all other organisms depend on them for their needs. Green plants carry out photosynthesis, a physico chemical process by which they use the light energy to drive the synthesis of organic compounds. Ultimately, all living forms on Earth depend on sunlight for energy. The use of energy from the sunlight by the plants doing photosynthesis is the basis of life on Earth. Photosynthesis is important due to two reasons. It is the primary source of all the food on the earth. It is also responsible for the release of oxygen into the atmosphere by the green plants. Have you ever thought what would happen if there were no oxygen to breathe? This chapter focuses on the structure of the photosynthetic machinery and the various reactions that transform the light energy into chemical energy. What do we know? Let us try to find out what we already know about photosynthesis. Some simple experiments you may have done in the earlier classes have shown that chlorophyll, green pigment of the leaf, light, and carbon dioxide are required for photosynthesis to occur. You may have carried out the experiment to look for starch formation in two leaves, a variegated leaf or a leaf that was partially covered with black paper and the one that was exposed to light. On testing these leaves for starch, it was clear that photosynthesis occurred only in the green parts of the leaves in the presence of the light. Another experiment you may have carried out is the half-leaf experiment, where a part of the leaf is enclosed in a test tube containing some KOH-soaked cotton, which absorbs carbon dioxide, while the other half is exposed to air. The setup is then placed in light for some time. On testing for starch later in the two halves of the leaf, you must have found that the exposed part of the leaf tested positive for starch, while the portion that was in the tube tested negative. This showed that carbon dioxide was required for photosynthesis. Can you explain how this conclusion could be drawn? Early experiments. It is interesting to learn about those simple experiments that led to the gradual development in our understanding of photosynthesis. Joseph Priestley from 1733 to 1804 in 1770 performed a series of experiments that revealed the essential role of air in the growth of green plants. Priestley, you may recall, discovered oxygen in 1774. Priestley observed that a candle burning in a closed space, a bell jar, soon gets extinguished. Similarly, a mouse would soon suffocate in a closed space. He concluded that the burning candle or animal that breathed the air both somehow damaged the air. But when he placed a mint plant in the same bell jar, he found that the mouse stayed alive and the candle continued to burn. Priestley hypothesized as follows: plants restore to the air whatever the breathing animals and the burning candles remove. Can you imagine how Priestley would have conducted the experiment using a candle and a plant? Remember, he would need to rekindle the candle to test whether it burns after a few days. How many different ways can you think of to light the candle without disturbing the setup? Using a similar setup as the one used by Priestley, but by placing it once in the dark and once in the sunlight, Jan Ingenhouse from 1730 to 1799 showed that sunlight is essential to the plant process that somehow purifies the air fouled by the burning candles or breathing animals. Ingenhouse, in an elegant experiment with an aquatic plant, showed that in bright sunlight small bubbles were formed around the green parts, while in the dark they did not. Later, he identified these bubbles to be of oxygen. Hence, he showed that it is only the green parts of the plant that could release oxygen. It was not until about 1854 that Julius von Sachs provided evidence for the production of glucose when plants grow. Glucose is usually stored as starch. His later studies showed that the green substance in the plant's chlorophyll, as we know it now, is located in special bodies later called chloroplast within the plant cells. He found that the green parts in the plants is where the glucose is made and that the glucose is usually stored as starch. Now consider the interesting experiments done by T.W. Engelmann from 1843 to 1909. Using a prism, he split light into its spectral components and then illuminated a green algae cladophora placed in the suspension of aerobic bacteria. The bacteria were used to detect the sites of O2 evolution. He observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum. The first action spectrum of photosynthesis was thus described. It resembles roughly the absorption spectrum of chlorophyll A and B. By the middle of the 19th century, the key features of the plant photosynthesis were known, namely that plants could use the light energy to make carbohydrates from carbon dioxide and water. The empirical equation representing the total process of photosynthesis for oxygen-evolving organisms was then understood as CO2 plus H2O in the presence of light gives CH2O plus O2, where CH2O represented a carbohydrate, example glucose, a 6-carbon sugar. A milestone contribution to the understanding of the photosynthesis was that made by the microbiologist Cornelius van Niel from 1897 to 1985, who, based on his studies of purple and green bacteria, demonstrated that photosynthesis is essentially a light dependent reaction in which hydrogen from a suitable oxidizable compound reduces the carbon dioxide to carbohydrates. This can be expressed by 2H2A plus CO2 in the presence of light gives 2A plus H2O plus CH2O. In green plants, H2O is the hydrogen donor and is oxidized to O2. Some organisms do not release the oxygen during photosynthesis. When H2S instead is the hydrogen donor for purple and green sulfur bacteria, the oxidation product is sulfur or sulfate depending on the organism and not O2. Hence, he inferred that the O2 evolved by the green plants comes from H2O, not from carbon dioxide. This was later proved by using radioisotopic techniques. The correct equation that would represent the overall process of photosynthesis is therefore 6CO2 plus 12H2O in the presence of light gives 6 C6H12O6 plus 6H2O plus 6O2, where C6H12O6 represents glucose. The O2 released is from water. This was proved using radioisotope techniques. Note that this is not a single reaction but description of a multi-step process called photosynthesis. Can you explain why 12 molecules of water as substrate are used in the equation given below? Where does photosynthesis takes place? You would of course answer in the green leaf, or you may add in the chloroplast based on what you have earlier read in chapter 8. You are definitely right. Photosynthesis does take place in the green leaves of the plants, but it does so also in the other green parts of the plants. Can you name some other parts where you think photosynthesis may occur? You would recollect from previous unit that the mesophyll cells in the leaves have a large number of the chloroplast. Usually the chloroplast align themselves along the walls of the mesophyll cells such that they get the optimum quantity of the incident light. When do you think the chloroplast will be aligned with their flat surfaces parallel to the walls? When would they be perpendicular to the incident light? You have studied the structure of chloroplast in chapter 8. Within the chloroplast, there is the membranous system consisting of grana, the stroma lamellae, and the fluid stroma. There is a clear division of the labor within the chloroplast. The membrane system is responsible for trapping the light energy and also for the synthesis of ATP and NADPH. In stroma, enzymatic reactions incorporate carbon dioxide into the plant, leading to the synthesis of sugar, which in turn forms starch. The former set of reactions, since they are directly light-driven, are called the light reactions. The latter are not directly light-driven but are dependent on the products of light reactions, ATP and NADPH. Hence, to distinguish the latter, they are called by convention as dark reactions. However, this should not be construed to mean that they occur in darkness or that they are not light dependent. How many pigments are involved in photosynthesis? Looking at the plants, have you ever wondered why and how there are so many shades of greens in their leaves, even in the same plant? We can look for an answer to this question by trying to separate the leaf pigments of any green plant through paper chromatography. A chromatographic separation of the leaf pigment shows that the color that we see in the leaves is not due to a single pigment but due to 4 pigments: chlorophyll A, bright or blue-green in the chromatogram; chlorophyll B, which is yellow-green; xanthophylls yellow; and carotenoids yellow to yellow-orange. Let us now see what roles various pigments play in photosynthesis. Pigments are the substances that have an ability to absorb the light at specific wavelengths. Can you guess which is the most abundant plant pigment in the world? Let us study the graph showing the ability of the chlorophyll A pigment to absorb light of different wavelengths. Of course, you are familiar with the wavelengths of the visible spectrum of light as well as of Vibgyor. From figure 13.3a, can you determine the wavelength color of the light at which the chlorophyll A shows the maximum absorption? Does it show any other absorption peak at any other wavelengths too? If yes, which one? Now look at figure 13.3b, showing the wavelengths at which maximum photosynthesis occurs in a plant. Can you see that the wavelengths at which there is maximum absorption by chlorophyll A, that is in the blue and the red regions, also shows higher rate of photosynthesis. Hence, we can conclude that chlorophyll A is the chief pigment associated with photosynthesis. But by looking at figure 13.3c, can you say that there is a complete one-to-one overlap between the absorption spectrum of chlorophyll A and the action spectrum of photosynthesis? These graphs together show that most of the photosynthesis takes place in the blue and the red regions of the spectrum. Some photosynthesis does take place at the other wavelengths of the visible spectrum. Let us see how this happens. Though chlorophyll is the major pigment responsible for trapping light, other thylakoid pigments like chlorophyll B, xanthophylls, and carotenoids, which are called accessory pigments, also absorb light and transfer the energy to chlorophyll A. Indeed, they not only enable a wider range of wavelength of incoming light to be utilized for photosynthesis, but also protect the chlorophyll A from photooxidation. What is light reaction? Light reactions of the photochemical phase include light absorption, water splitting, oxygen release, and the formation of high energy chemical intermediates ATP and NADPH. Several complexes are involved in the process. The pigments are organized into two discrete photochemical light harvesting complexes LHC within the photosystem 1 PS1 and the photosystem 2 PS2. These are named in the sequence of their discovery and not in the sequence in which they function during the light reaction. The LHC are made up of hundreds of pigment molecules bound to proteins. Each photosystem has all the pigments except one molecule of chlorophyll A, forming a light harvesting system also called antennae. These pigments help to make photosynthesis more efficient by absorbing different wavelengths of light. A single chlorophyll A molecule forms the reaction center. The reaction center is different in both the photosystems. In PS1, the reaction center chlorophyll A has an absorption peak at 700 nanometer, hence is called P700, while in PS2 it has an absorption maxima at 680 nanometer and is called P680. The electron transport. In photosystem 2, the reaction center chlorophyll A absorbs 680 nanometer wavelength of red light, causing electrons to become excited and jump into an orbit farther from the atomic nucleus. These electrons are picked up by an electron acceptor, which passes them onto an electron transport system consisting of cytochromes. This movement of the electrons is downhill in terms of an oxidation reduction or redox potential scale. The electrons are not used up as they pass through the electron transport chain but are passed on to the pigments of the photosystem PS1. Simultaneously, electrons at the reaction center of the PS1 are also excited when they receive red light of wavelength 700 nanometer and are transferred to another acceptor molecule that has a greater redox potential. These electrons then are moved downhill again, this time to a molecule of energy-rich NADP+. The addition of these electrons reduces NADP plus to NADPH plus H plus. This holds a scheme of transfer of electrons starting from the PS2 uphill to the acceptor, down the electron transport chain to PS1, excitation of electrons transferred to another acceptor, and finally downhill to NADP plus, causing it to be reduced to NADPH plus H plus, is called as the Z scheme due to its characteristic shape. This shape is formed which when all the carriers are placed in the sequence on the redox potential scale. Splitting of water. You would then ask, how does PS2 supply electrons continuously? The electrons that were removed from the photosystem 2 must be replaced. This is achieved by electrons available due to splitting of water. The splitting of water is associated with the PS2. Water is split into H+, nascent oxygen and electrons. This creates oxygen, one of the net products of photosynthesis. The electrons needed to replace those removed from the photosystem 1 are provided by photosystem 2. 2H2O splits to give 4H plus O2 plus 4 electrons. We need to emphasize here that the water splitting complex is associated with the PS2, which itself is physically located on the inner side of the membrane of the thylakoid. Then, where are the protons and O2 formed likely to be released? In the lumen or on the outer side of the membrane. Cyclic and non-cyclic photophosphorylation. Living organisms have the capability of extracting energy from oxidizable substances and store this energy in the form of bond energy. Special substances like ATP carry this energy in their chemical bonds. The process of which ATP is synthesized by the cells in mitochondria and chloroplast is named phosphorylation. Photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate in the presence of light. When the two photosystems work in a series, first the PS2 and then the PS1, a process called non-cyclic photophosphorylation occurs. The two photosystems are connected to an electron transport chain as seen earlier in the Z scheme. Both ATP and NADPH plus H plus are synthesized by this kind of electron flow. When only PS1 is functional, the electron is circulated within the photosystem and the phosphorylation occurs due to the cyclic flow of electrons. A possible location where this could be happening is in the stroma lamellae. While the membrane of the lamellae of the grana have both PS1 and PS2, the stroma lamellae membranes lack PS2 as well as NADP reductase enzyme. The excited electrons does not pass on to NADP plus but is cycled back to the PS1 complex through the electron transport chain. The cyclic flow hence results only in the synthesis of ATP but not of NADPH plus H plus. Cyclic photophosphorylation also occurs when only light of wavelengths beyond 680 nanometer are available for excitation. Chemiosmotic hypothesis. Let us now try and understand how actually ATP is synthesized in the chloroplast. The chemiosmotic hypothesis has been put forward to explain the mechanism. Like in respiration, in photosynthesis too, ATP synthesis is linked to the development of a proton gradient across a membrane. This time, these are the membranes of the thylakoid. There is one difference though. Here, the proton accumulation is towards the inside of the membrane, that is, in the lumen. In respiration, proton accumulates in the intermembrane space of the mitochondria when electrons move through the electron transport system. Let us understand what causes the proton gradient across the membrane. We need to consider again the process that takes place during the activation of the electrons and their transport to determine the steps that cause a proton gradient to develop. A. Since splitting of the water molecules takes place on the inner side of the membrane, the protons or hydrogen ions are produced by the splitting of water accumulate within the lumen of the thylakoids. B. As electrons move through the photosystems, protons are transported across the membrane. This happens because the primary acceptor of electron, which is located towards the outer side of the membrane, transfers its electron not to an electron carrier, but to a hydrogen carrier. Hence, this molecule removes a proton from the stroma while transporting an electron. When this molecule passes on its electron to the electron carrier on the inner side of the membrane, the proton is released into the inner side or the lumen side of the membrane. C. The NADP reductase enzyme is located on the stroma side of the membrane. Along with the electrons that comes from the acceptor of electrons of PS1, protons are necessary for the reduction of NADP plus to NADPH plus H plus. These protons are also removed from the stroma. Hence, within the chloroplast, protons in the stroma decreases in number while in the lumen there is accumulation of protons. This creates a proton gradient across the thylakoid membrane as well as measurable decrease in the pH or in the lumen. Why are we so interested in the proton gradient? This gradient is important because it is the breakdown of this gradient that leads to the release of energy. The gradient is broken down due to the movement of the protons across the membrane to the stroma through the transmembrane channel of the F0 of the ATPase. The ATPase enzyme consists of two parts. One called the F0 is embedded in the membrane and forms the transmembrane channel that carries out facilitated diffusion of protons across the membrane. The other portion is called F1 and protrudes on the outer surface of the thylakoid membrane on the side that faces the stroma. The breakdown of the gradient provides enough energy to cause a conformational change in the F1 particle of the ATPase, which makes the enzyme synthesis several molecules of energy-packed ATP. Chemiosmosis requires a membrane, a proton pump, a proton gradient, and ATPase. Energy is used to pump the protons across a membrane to create a gradient or a high concentration of protons within the thylakoid lumen. ATPase has a channel that allows diffusion of the protons back across the membrane. This releases enough energy to activate the ATPase enzyme that catalyzes the formation of ATP. Along with the NADPH produced by the movement of the electrons, ATP will be used immediately in the biosynthetic reaction taking place in the stroma, responsible for fixing CO2 and synthesis of sugar. Where are the ATP and NADPH used? We learned that the products of the light reaction are ATP, NADPH and O2. Of these, O2 diffuses out of the chloroplast while ATP and NADPH are used to drive the process leading to the synthesis of food, more accurately sugars. This is the biosynthetic phase of photosynthesis. This process does not directly depend on the presence of light but is dependent on the products of the light reaction, that is ATP and NADPH, besides CO2 and H2O. You may wonder how this could be verified. It is simple. Immediately after light becomes unavailable, the biosynthetic process continues for some time and then stops. If then light is made available, the synthesis starts again. Can we hence say that calling the biosynthetic phase as the dark reaction is a misnomer? Discuss this amongst yourselves. Let us now see how the ATP and the NADPH are used in the biosynthetic phase. We saw earlier that CO2 is combined with H2O to produce CH2O or sugars. It was of interest to scientists to find out how this reaction proceeded, or rather, what was the first product formed when CO2 is taken into a reaction or fixed. Just after World War II, among the several efforts to put radioisotopes to beneficial use, the work of Melvin Kelvin is exemplary. The use of the radioactive C14 by him in algal photosynthesis studies led to the discovery of the first CO2 fixation product was a 3-carbon organic compound. He also contributed to working out the complete biosynthetic pathway, hence it was called the Kelvin cycle after him. The first product identified was 3-phosphoglyceric acid, or in short PGA. How many carbon atoms does it have? Scientists also tried to know whether all the plants have PGA as the first product of CO2 fixation or whether any other product was formed in other plants. Experiments conducted over a wide range of plants led to the discovery of another group of plants where the first stable product of CO2 fixation was again an organic acid but one which had 4 carbon atoms in it. This acid was identified to be oxaloacetic acid or OAA. Since then, CO2 assimilation during photosynthesis was said to be of two main types: those plants in which the first product of CO2 fixation is C3 acid PGA, that is the C3 pathway, and those in which the first product was a C4 acid OAA, that is the C4 pathway. These two groups of plants showed other associated characteristics that we will discuss later. The primary acceptor of CO2. Let us now ask ourselves a question that was asked by the scientists who were struggling to understand the dark reaction. How many carbon atoms would a molecule which have after accepting CO2 would have 3 carbons of PGA? The studies were unexpectedly showed that the acceptor molecule was a 5 carbon keto sugar. It was ribulose bisphosphate RUBP. Did any of you think of this possibility? Do not worry. The scientists also took a long time and conducted many experiments to reach this conclusion. They also believed that since the first product was a C3 acid, the primary acceptor would be a 2 carbon compound. They spent many years trying to identify a 2 carbon compound before they discovered the 5 carbon RUBP. The Kelvin cycle. Kelvin and his co-workers then worked out the whole pathway and showed that the pathway operated in a cyclic manner. The RUBP was regenerated. Let us now see how the Kelvin pathway operates and where the sugar is synthesized. Let us at the outset understand very clearly that the Kelvin pathway occurs in all photosynthetic plants. It does not matter whether they have C3 or C4 or any other pathways. For ease of understanding, the Kelvin cycle can be described under 3 stages: carboxylation, reduction, and regeneration. Carboxylation. Carboxylation is the fixation of CO2 into a stable organic intermediate. Carboxylation is the most crucial step of the Kelvin cycle where CO2 is utilized for the carboxylation of RUBP. This reaction is catalyzed by the enzyme RUBP carboxylase, which results in the formation of 2 molecules of 3-phosphoglyceric acid. Since this enzyme also has an oxygenation activity, it would be more correct to call it RUBP carboxylase oxygenase or Rubisco. Reaction. There are a series of reactions that lead to the formation of glucose. The steps involve utilization of 2 molecules of ATP for phosphorylation and 2 molecules of NADPH for reduction per CO2 molecule fixed. The fixation of the 6 molecules of the CO2 and 6 turns of the cycle are required for the removal of 1 molecule of glucose from the pathway. Regeneration. Regeneration of the CO2 acceptor molecule RUBP is crucial if the cycle is to continue uninterrupted. The regeneration steps require 1 ATP for phosphorylation to form RUBP. Hence, for every CO2 molecule entering the Kelvin cycle, 3 molecules of ATP and 2 of NADPH are required. It is probably to meet this difference in the number of ATP and NADPH used in the dark reaction that the cyclic phosphorylation takes place. To make 1 molecule of glucose, 6 turns of the cycle are required. Work out how many ATP and NADPH molecules will be required to make 1 molecule of glucose through the Kelvin pathway. It might help you to understand all of this if we look at what goes in and comes out of the Kelvin cycle. In 6 CO2 and out 1 glucose. For 18 ATP that goes in, 18 ADP comes out. For 12 NADPH that goes in, 12 NADP comes out. And the C4 pathway. Plants that are adapted to dry tropical regions have the C4 pathway mentioned earlier. Though these plants have the C4 oxaloacetic acid as the first CO2 fixation product, they use the C3 pathway or the Kelvin cycle as the main biosynthetic pathway. Then in what way are they different from the C3 plants? This is a question that you may reasonably ask. C4 plants are special. They have a special type of leaf anatomy. They tolerate higher temperatures and they show a response to high light intensities. They lack a process called photorespiration and have greater productivity of biomass. Let us understand these one by one. Study vertical sections of leaves of one of the C3 plant and other of the C4 plant. Did you notice the differences? Do both have the same type of mesophils? Do they have similar cells around the vascular bundle sheath? The particularly large cells around the vascular bundles of the C4 pathway plants are called the bundle sheath cells, and the leaves which have such anatomy are said to have Kranz anatomy. Crans means breadth and is a reflection of the arrangement of cells. The bundle sheath cells may form several layers around the vascular bundles. They are characterized by having a large number of chloroplasts, thick wall impervious to gaseous exchange, and no intercellular spaces. You may like to cut a section of the leaves of the C4 plants, maize or sorghum, to observe the crans anatomy and the distribution of mesophyll cells. It would be interesting for you to collect leaves of diverse species of plants around you and cut vertical sections of the leaves. Observe under the microscope. Look for the bundle sheath around the vascular bundles. The presence of the bundle sheath would help you identify the C4 plants. Now study the pathway in figure 13.9. This pathway that has been named the Hatch-and-Slack pathway is again a cyclic process. Let us study the pathway by listing the steps. The primary CO2 acceptor is a 3-carbon molecule phosphoenol pyruvate PEP and is present in the mesophyll cells. The enzyme responsible for this fixation is PEP carboxylase or PEP-case. It is important to register that the mesophyll cells lack Rubisco enzyme. The carbon acid oxaloacetic acid is formed in the mesophyll cells. It then forms other 4-carbon compounds like malic acid or aspartic acid in the mesophyll cells itself, which are transported to the bundle sheath cells. In the bundle sheath cells, these C4 acids are broken down to release CO2 and a 3-carbon molecule. The 3-carbon molecule is transported back to the mesophyll cells where it is converted to PEP again, thus completing the cycle. The CO2 released in the bundle sheath cells enter the C3 or the Kelvin pathway, a pathway common to all plants. The bundle sheath cells are rich in an enzyme, rubulose biphosphate carboxylase oxygenase Rubisco, but lack PEP-case. This is the basic pathway that results in the formation of the sugars. The Kelvin pathway is common to the C3 and the C4 plants. Did you note that the Kelvin pathway occurs in all the mesophyll cells of the C3 plants? In the C4 plants, it does not take place in the mesophyll cells but does so only in the bundle sheath cells. Photorespiration. Let us try and understand one more process that creates an important difference between C3 and C4 plants, photorespiration. To understand photorespiration, we have to know a little bit more about the first step of the Kelvin pathway, the first CO2 fixation step. This is the reaction where the RuBP combines with CO2 to form 2 molecules of 3-phosphoglyceric acid that is catalyzed by Rubisco. RuBP plus CO2 in the presence of Rubisco gives 2 molecules of 3-phosphoglyceric acid. Rubisco, that is the most abundant enzyme in the world. Do you wonder why? Is characterized by the fact that its active site can bind to both CO2 and O2, hence the name. Can you think how this could be possible? Rubisco has a much greater affinity for CO2 than for O2. Imagine what would happen if this were not so. This binding is competitive. It is the relative concentration of the O2 and the CO2 that determine which of the two will bind to the enzyme. In C3 plants, some O2 does bind to Rubisco and hence CO2 fixation is decreased. Here the RuBP, instead of being converted to 2 molecules of PGA, binds with O2 to form 1 molecule of phosphoglycerate and phosphoglycolate in the pathway called photorespiration. In the photorespiratory pathway, there is neither synthesis of sugar nor of ATP. Rather, it results in the release of CO2 with the utilization of ATP. In the photorespiratory pathway, there is no synthesis of ATP or NADPH. Therefore, photorespiration is a wasteful process. In C4 plants, photorespiration does not occur. This is because they have a mechanism that increases the concentration of CO2 at the enzyme site. This takes place when the C4 acid from the mesophyll is broken down in the bundle sheath cells to release CO2. This results in increasing the intercellular concentration of CO2. In turn, this ensures that the Rubisco functions as a carboxylase, minimizing the oxygenase activity. Now that you know that the C4 plants lack photorespiration, you probably can understand why productivity and yields are better in these plants. In addition, these plants show tolerance to high temperatures. Based on the above discussion, can you compare plants showing the C3 and the C4 pathway? Use the table format given and fill in the information. Factors affecting photosynthesis. An understanding of the factors that affect photosynthesis is necessary. The rate of photosynthesis is very important in determining the yield of the plants, including the crop plants. Photosynthesis is under the influence of several factors, both internal, plant and external. The plant factors include the number, size, age and orientation of the leaves, mesophyll cells and chloroplast, internal CO2 concentration and the amount of chlorophyll. The plant or the internal factors are dependent on the genetic predisposition and the growth of the plant. The external factors would include the availability of the sunlight, temperature, CO2 concentration and water. As a plant photosynthesis, all these factors will simultaneously affect its rate. Hence, though several factors interact and simultaneously affect photosynthesis or CO2 fixation, usually one factor is the major cause or is the one that limits the rate. Hence, at any point, the rate will be determined by the factor available at suboptimal levels. When several factors affect any biochemical process, Blackman's 1905 law of limiting factors comes into effect. This states the following. If a chemical process is affected by more than one factor, then its rate will be determined by the factor which is nearest to its minimal value. It is the factor which directly affects the process if its quantity is changed. For example, despite the presence of green leaf and optimal light and CO2 conditions, the plant may not photosynthesize if the temperature is very low. This leaf is given the optimal temperature, it will start photosynthesizing. Light. We need to distinguish between light quality, light intensity, and the duration of exposure to light while discussing light as a factor that affects photosynthesis. There is a linear relationship between the incident light and the CO2 fixation rates at low light intensities. At higher light intensities, gradually the rate does not show further increase as other factors become limiting. What is interesting to note is that light saturation occurs at 10% of the full sunlight. Hence, except for plants in shade or in the dense forest, light is rarely a limiting factor in nature. Increase in incident light beyond a point causes the breakdown of the chlorophyll and a decrease in photosynthesis. Carbon dioxide concentration. Carbon dioxide is the major limiting factor for photosynthesis. The concentration of CO2 is very low in the atmosphere, between 0.03 and 0.04%. Increase in the concentration up to 0.05% can cause an increase in CO2 fixation rates. Beyond this level can become damaging over longer periods. The C3 and the C4 plants respond differently to CO2 concentrations. At low light conditions, neither group responds to high CO2 conditions. At high light intensities, both C3 and the C4 plants show increase in the range of photosynthesis. What is important to note is that the C4 plants show saturation at about 360 microliters per liter, while the C3 plants respond to increased CO2 concentration and saturation is seen only beyond 450 microliter per liter. This current availability of CO2 levels is limiting to the C3 plants. The fact that the C3 plants respond to higher CO2 concentration by showing increased rate of photosynthesis leading to higher productivity has been used for some greenhouse crops such as tomatoes and the bell pepper. They are allowed to grow in carbon dioxide enriched atmosphere, but that leads to higher yields. Temperature. The dark reactions being enzymatic are temperature controlled. Though the light reactions are also temperature sensitive, they are affected to a much lesser extent. The C4 plants respond to higher temperatures and show higher rates of photosynthesis, while the C3 plants have a much lower temperature optimum. The temperature optimum for photosynthesis of different plants also depends on the habitat that they are adapted to. Tropical plants have a higher temperature optimum than the plants adapted to temperate climates. Water. Even though water is one of the reactants in the light reaction, the effect of water as a factor is more through its effect on the plants rather than directly on photosynthesis. Water stress causes the stomata to close, hence reducing the CO2 availability. Besides, water stress also makes leaves wilt, thus reducing the surface area of the leaves and their metabolic activity as well. Summary. Green plants make their own food by photosynthesis. During this process, carbon dioxide from the atmosphere is taken in by the leaves through stomata and used for making carbohydrates, principally glucose and starch. Photosynthesis takes place only in the green parts of the plants, mainly the leaves. Within the leaves, the mesophyll cells have a larger number of chloroplasts that are responsible for CO2 fixation. Within the chloroplast, the membranes are sites of the light reaction, while the chemiosmotic pathway occurs in the stroma. Photosynthesis has two stages, the light reaction and the carbon-fixing reactions. In the light reaction, the light energy is absorbed by the pigments present in the antenna and funneled to the special chlorophyll A molecules called reaction center chlorophylls. There are two photosystems, PS1 and PS2. PS1 has a 700 nanometer absorbing chlorophyll A P700 molecule at its reaction center, while PS2 has a P680 reaction center that absorbs red light at 680 nanometer. After absorbing light, electrons are excited and transferred through the PS2 and PS1 and finally to NAD, forming NADH. During this process, a proton gradient is created across the membrane of the thylakoid. The breakdown of the proton gradients due to the movement through the F0 part of the ATPase enzyme releases enough energy for synthesis of ATP. Splitting of water molecules is associated with the PS2, resulting resulting in the release of O2, protons, and transfer of electrons to PS2. In the carbon fixation cycle, carbon dioxide is added by the enzyme Rubisco to a 5-carbon compound RUBP that is converted to 2 molecules of 3-carbon PGA. This is then converted to sugar by the Kelvin cycle and the RUBP is regenerated. During this process, ATP and NADPH synthesized in the light reaction are utilized. Rubisco also catalyzes a wasteful oxygenation reaction in C3 plants called photorespiration. Some tropical plants show a special type of photosynthesis called the C4 pathway. In these plants, the first product of CO2 fixation that takes place in the mesophyll is a 4-carbon compound. In the bundle sheet cells, the Kelvin pathway is carried out for the synthesis of carbohydrates. Thank you.